From Microbe TV, this is Immune, episode number 14, recorded on November 13th, 2018. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again. In Ithaca, we've got some snow, so winter's here.
No snow here, but it's pouring. Pouring rain all night.
I don't know which is worse.
Yeah. Either way, it's driving is harder.
Yeah.
Also joining us from Worcester, Ohio, Steph Langel. Dr. Steph Langel.
Hey there. Hey, it's good to be on the other side. I have to say, this is, there's no visual for this, but I'm doing a little dance here to celebrate my post-dissertation.
We're dancing for you too.
Oh, thank you.
So
thank you.
Steph
Yes.
defended last week, right?
Yes, last Wednesday. It was on Halloween. Or 2 Wednesdays ago. It would have been, 2 Wednesdays ago now.
How did it go?
Time goes by fast. It went by well. I mean, the presentation, presentations and me, we usually do well. I do enjoy speaking. And by that point, I had presented this data a lot of different times. So I felt comfortable. The questions were engaging and I could answer them. Then, of course, the defense is multiple hours spread out afterwards. And, to be expected, there was, because your data so well to that point, you can answer a lot of the questions. And I don't know, you all can tell me what you experience with your PhD students, but you're so focused on this research and this field. And so then, trying to answer the questions when it comes out of the scope of that field. And I think there was just a few questions that I had to pause and really kind of work my way through, think about the mechanisms. Okay, how could this affect, this other immune response? And so I think that's probably the transition from like PhD to postdoc to PI is really, okay, this area, but you need to start putting it in the building blocks of biology. So yeah, it went really well. And I'm just happy I'm on this side of things.
Yeah, it's no better on this side, I have to tell you.
Let me live in this moment, Vincent, for a little while.
Okay.
Right? No, just kidding.
And in January, you're going to be moving. That's exciting.
Yes, I'll be moving to Duke University, and I will be starting a postdoc with Dr. Sally Permar and the Human Vaccine Institute. And we're going to be working in maternal immunology and really trying to understand how to develop vaccines during pregnancy and to boost lactogenic immunity or milk immunity. So it's a similar field of what I'm doing now, but we're going to focus kind of on more mechanistic. I was doing swine research, so it's more vaccinology, and we're going to be looking more mechanistically at these receptors in the mammary gland and how they can transport immunoglobulins and maybe even cells to the milk and then to the neonate and what that response is. So,
what are you doing that in animals? What humans?
We're going to do it in mouse, in a mice model. Mm-hmm.
They'll be easier for you to pick up.
Oh, let me tell you. Oh, my goodness. I will be looking forward to now. Of course, we're going over milking protocols, and the amount of milk you can get from a mouse is about 100. And I mean, you're like pressing these little teats to get like 150 microliters. So it'll be a difference, but I'm excited.
I always had this vision of you wrestling huge pigs. And on Instagram the other day, someone posted a video of this guy somewhere, and there were all these big pigs, and he ran up behind one and he jumped on it, and the pig then twirled in circles and then ran off. And I said, "That's Steph."
Yeah, I think Ayakuk would be mad at me if I'm riding my pig. Yeah, no, they're very large animals, so it can be interesting. Yeah. Mice models will be a different kind of life, but I'm excited.
Cool.
It'll be good.
Well, congratulations.
Thanks, guys, and thanks for your support. I definitely contribute, you both and this podcast, for helping me improve my communication skills, which really helped in the defense.
And hopefully Duke will allow you to continue podcasting.
I would think so. I mean, we do this once a month, and I got to be reading papers anyways. So, might as well do a podcast.
At some point, we'll get your boss on, right?
That would be great. She does a lot of HIV transmission in milk is kind of the cornerstone in preventing that transmission of her lab.
Yeah.
But they do CMV, Zika, and then I'll be doing the Rota.
That'll be fine. We'll give you about 6 months to a year, and then we'll tell you, okay, now get her on.
Yeah, we'll give her some time.
Yeah, she'll hear a few episodes and realize how cool it is, right?
Of course.
All right, cool. Okay, this week, Cindy promised to explain everything about this year's physiology or medicine Nobel Prizes.
I don't know about everything, but I will do my best.
You starting where both winners were born. I'm just kidding. You probably could say one's
in Japan.
Yeah, you know James Allison used to be in New York City.
Yes, I do.
But I never saw him, not once.
No? Oh. Yeah, he grew up in Texas, I think.
Yeah, I think that's right. Yeah.
Yeah. So, so the 2018 Nobel Prize in Physiology or Medicine was awarded to two individuals, James Allison and Tasuku Honjo, for their work on checkpoint blockade as a treatment for cancer. So there's a lot to talk about related to this and how the immune system works, how T cells kill cancer, and why on earth these things called checkpoint blockades actually have such a high therapeutic activity in human cancer. And just as a note to who these two individuals are, I won't give their entire life history. But yeah, Jim Allison is currently chairman of immunology at the University of Texas, the MD Anderson Cancer Center in Houston. And he is known for a number of things, but he is the discoverer of a molecule called CTLA4. And he also did the initial experiments that showed that this could be developed as a cancer immunotherapeutic. So he generated antibodies to this molecule and showed that if they treated animals with those antibodies, those animals were then able to eliminate their cancers. And the other individual, Tasuku Honjo, who is at Kyoto University, and he discovered a molecule called PD-1. And it is these two molecules, CTLA4 and PD-1, that are being used that are being targeted in human cancer. And the precursor to this, and interestingly, we can go into this a little bit, is that Jim Allison, Tasuku Honjo, as well as Liping Chen, Arlene Sharp, and Gordon Freeman all shared this prize called the Warren Alpert Foundation Prize last year. And there's some controversy over this because all of those individuals contributed, and even an additional individual contributed to the fundamental discoveries that led to what was awarded for this Nobel Prize to only two individuals. And so it's kind of an interesting thing because the Nobel Prize, you may or may not know, can only be given to a maximum of 3 individuals. And so I just wonder, with the increase in interdisciplinary science, how is this going to affect the selection of Nobel Prize winners? Because as more and more people are involved in the fundamental research for the discoveries, how do you choose who actually is awarded the prize for those discoveries?
Well, discoveries are never made just by the people who get the prizes, right? You always have a big lab behind them. So in a way, it's already not fair. And it reminds me of the Nobel in the early 50s given to John Enders, who figured out you could grow poliovirus in cell cultures for the first time. And they were going to give it to him. He said, no, I have 2 people. And one of them was a MD fellow, and maybe the other was a student. I'm not even sure. He said they did the work. And so Enders, Weller, and Robbins ended up getting the prize. So sometimes the recipient can say, no, it's not fair just to give it to me. But I think most people don't. They just say, no, okay, I'll take it, right?
Okay, yeah. Yeah, I think I don't think the Nobel is going to go away. So I think it's just going to be more controversy. And I mean, I do like the Nobel because it seems to be one of the awards that the public really, their ears prick up. They like to be engaged in, okay, who won the Nobel Prize? But it doesn't really give a big full picture of how science works. So I don't know,
That's a good point.
Your feeling about it.
That's a good point about engaging the public, because it certainly does, right? Because it gets big,
more
it gets
than any
media.
other award.
Totally, it gets big media attention.
Right.
Except maybe the one that Jennifer Doudna got a couple of years ago where you get over $1 million, What is that? The
the Lasker?
Not
no,
the last.
Not the. Oh, I don't. I don't remember.
It is the Breakthrough Prize.
I was going to say Discovery Prize, Breakthrough Prize. Yeah.
Yeah, she got it with Charpentier, and that also gets attention because it's a lot of money. But there's nothing like a Nobel. But there are a lot of people have published articles on why the Nobels are actually not good for science. Right.
Right.
So, but you're right. They're not going away.
Right.
But I think, as part of the science communication part of it, I think that, they're raising the public awareness of these scientific discoveries. Mm-hmm. And that really does benefit science.
Yeah, I agree.
And in this case, we're going to talk about this, the 2018 prize. And this is really something that, your everyday individual on the street is probably going to encounter sometime in their life, because 1 out of 4 people have cancer. Right. This is going to become the standard of care for a number of cancers.
Yep.
Yeah.
And I might point out that traditionally American Nobels get invited to the White House, but that hasn't happened for the past 2 years.
Interesting.
We have a Nobel here, Joachim Frank, who got it in 2017 for cryo-EM.
Oh, yeah, that's right.
He said, I hope I don't get invited to the White House because I don't want to go.
Oh, boy.
Oh.
And he didn't.
That's kind of...
It's very sad. It's sad. Science is a big part of our society, which is led by the president, and science should be part of that, too, and it's too bad that it's not.
That's true.
I'm not making any statements. I'm just saying it's too bad.
It is.
Because that further, emphasizes to the public, hey, this is great stuff, that these scientists have won this from the United States and we're going to celebrate them and so forth. And that's not done anymore. It's too bad.
Well, if we want to talk about pride of country, I don't mean to digress further, but I mean, an American Nobel, why not? That's, a definition of pride of country. So,
yeah. So, we still do pretty well in terms of Nobels, right? And so we should be proud of the scientific enterprise here. Other countries do well. So Japan is great for a small country. They get a lot of Nobels. I think it should be a matter of national pride.
It should be. When we shortchange those major prizes in the scientific discoveries, we're shortchanging advancement of science.
Yeah. But I don't think, sports teams often get invited, musicians. I don't think anyone's gone to the White House the last couple of years. So it's not just picking on science. It's
right. Yeah. Well, I think, this particular discovery is really exciting because I think it's one of the best examples of this idea of translational biology. Because these were basic research studies on trying to understand how the immune system works. And when they were working on this 20 years ago, there was no prediction of what future applications these were going to have. And it was just when they started to uncover the fundamentals and sat back and thought about, what could we do with this, that the idea of trying these in cancer arose. And without that we wouldn't be where we are, and you'll see as we go through the next hour or so how amazing these immunotherapies are for treatment of cancer. And I wanted to make a note because I was researching, there's a lot out on the internet now about the individuals who won this and, their history and all of this. And so I was watching some of this, and Allison himself says he never envisioned the success of this because, he really was interested in the fundamentals. But he is very specific about an interesting and important barrier to progress in this area, which I hadn't thought about and I think is interesting because the Cancer Research Institute was doing an interview with him, and they asked him, what do you see as the major barriers to progress in this field, of cancer immunotherapy? And he responded, one of the barriers that frustrates many of the people in the field is that the different drugs, which could be used in combination, are sometimes held by different companies. And so, because then it's for very difficult reasons that have nothing to do with science, everything to do with business, it's hard to get the combination therapies in place. And when I was watching some of the talks he's given, what he's showing now, and we'll get into this a little bit, is these two molecules with CTLA4 and PD1, each alone are not very effective, but when you put them together, they're incredibly effective. And so if one company owns one drug and one company owns the other, how do you get the two of them together to be able to do these clinical trials? Because they invest a lot of money in these things, and they intend to make a lot of money off of these blockbuster drugs. But yet, if they're in by themselves as solo therapies, they don't work very well. So it's kind of an interesting question.
That cannot hold back therapies, right? That's crazy.
Exactly. Exactly.
I hope that change. You right here, it's beginning to change. I hope it does, because it's all about saving lives, right? It's not about.
It should be.
The primary thing is not making money, although I understand the companies have to do that. It should be about changing people's lives.
I agree.
Well, it may be more with him talking about it and raising awareness than more the public is.
Yeah.
They're maybe start to say, "Hey, what's going on here?"
Yeah.
Drive change.
Yeah.
Or someday, people, why can't I have that drug? Well, two companies are fighting over it, so it's not available. Enough people have that said to them. And actually, it only needs one very rich person to be told you can't have the drug because two companies were fighting over it, and then it'll affect change.
Right.
Yeah. Unfortunately, that might actually have to happen. But I do know that they have managed to do clinical trials with drugs against these two molecules. So there is progress in that area. But so today what I wanted to do is talk about a whole bunch of different things related to this, but so that everybody can understand what these immunotherapies really are and why it's so important. I wanted to introduce a little bit about T cells and how they're working so that we can understand how they can be shut off and why these immunotherapies or things called checkpoint inhibitors do what they do. And so I think we've mentioned it before, but remember, T cells are these cells in the immune system that can go and kill target cells. So there's many different flavors of these things. CD8 T cells are particularly good at recognizing either infected cells or cancer cells, and they can deploy a variety of different weapons against those cells to kill them. But in order to do that, they need to have a couple of different signals, because just unleashing them is very toxic. So they have strict control mechanisms, and we call these signal 1, signal 2. And so what T cells and T cell receptors recognize are little bits of either cells or microorganisms presented and held kind of like a hot dog in a bun in a protein called an MHC molecule. And so when the T cells see that, they have a T cell receptor that specifically recognizes that bit. And that match has to be perfect. Otherwise, it doesn't work. But that match alone is not enough. And so you need this signal 2. And signal 2 is something that tells the T cell what you're seeing is actually something you should respond to. And so it's really only the signal 2 only happens really in the context of danger or infection. And so that's a way in which this presentation of this fragment or peptide in the context of MHC can be interpreted as something that the T cell should respond to. And then the third thing, there's a third signal, that's cytokines that shape the type of T cell that you get out. So is it particularly good at inducing antibody responses in B cells, or is it particularly good in attacking other cells? And so those soluble molecules are in signal 3 are what shapes that type of immune response. And so when T cells are activated, you can't just leave them go with this positive stimulus out into the wild. So these T cells also at the same time sort of upregulate dampening molecules or inhibitory molecules. So, and this allows the immune system to control the level of T cell response. And that's really important. So it's like fine-tuning the response. The more negative signals or suppressor molecules you upregulate, the less that T cell is going to be activated. And so depending on the magnitude of the response, you might have more or less of that. But these molecules are really important for shutting down the response and dampening and tampering the response.
Same kind of theme as last time, where we talked about a long non-coding RNA that regulates innate responses.
Right, exactly.
Right.
So that idea of tuning immune responses is so important in so many different contexts that we're understanding now. It's not a flip. It's not flipping a switch. It's not an on or an off. It's a rheostat, right? So it's fine tuning the level of response in many different ways. And so the other way in which these molecules that we're going to talk about are important. So these molecules, this PD-1 and CTLA-4, are two of these molecules that are these dampening molecules that get upregulated when a T cell is activated. The other place we know these are really important is maintaining tolerance to our own body. So preventing autoimmune disease. So when the body generates the many different T cell receptors that are expressed on the variety of different T cells in the body. There needs to be a way to determine whether or not those T cell receptors are recognizing self. And this all happens in the thymus during this process called selection. And so in the thymus, if the T cells recognize something from your own body with high specificity, they get deleted. And that's called negative selection. And they only make it out of the thymus if they can recognize something. If they don't have a properly made T cell receptor, they don't make it out of the thymus. And so that's positive selection. So these T cells have to make it through this gauntlet of selection processes in the thymus. But if you think about it, again, everything is a continuum. There's not totally self-reactive cells and totally non-self-reactive cells. There's going to be some that are, where do you draw that line, right? So there's some of those that are going to be somewhat autoreactive that are going to make it out into the periphery. And this is contrary to what was taught in immunology for a long time. We said if they react with self, they never make it out of the thymus, they get deleted. But the reality is that some semi-autoreactive cells make it out of the thymus. And one wonders if maybe part of that is responsible for cancer immunosurveillance, right? Because cancer cells are actually self.
Yeah.
So if we have those T cells out there, we have the potential to attack those cancer cells in the future if we needed them. But those cells that make it out into the periphery that can recognize some level of self get shut down. That's a tolerance. We don't want them active all the time. So here's the conundrum. We have them, but they're mostly suppressed. So how do you activate them against a cancer cell? Right. So that's a fundamental problem that we have. So this idea of peripheral tolerance or shutting down those cells that make it out of the gauntlet of the thymus and still have some autoreactivity. So how is that tolerance mediated? So how do we prevent T cells that might be capable of recognizing a host cell from always attacking cells in the body? And this happens because we have these molecules that are expressed on T cells that can engage cells engage molecules on self that dampen those T cell responses. And two of those, as you might imagine, are PD-1 and CTLA-4. So if you make mice that are deficient in either one of these, they get overwhelming autoinflammatory diseases. So clearly we know that these are restricting the ability of these autoreactive T cells to go out and attack self. We also know from studies that were done a number of years ago that cancer cells have gotten really smart. Now I'm anthropomorphizing cancer cells here, right? But they figured out this whole system and they said, well, what if I put the ligands on the surface of myself, speaking from the cancer cell, that shuts those T cells down. So now, in addition to this general peripheral tolerance, even if they were capable of attacking the cancer cell has now upregulated the ligands for these molecules and used them to shut down the T cell responses within a tumor. So even if T cells were primed and capable of killing the tumor cell, they get in the tumor and the cancer cell shuts them down.
What a betrayal. I always think of cancer as just such a betrayal of your own body.
And I wanted to give you a quote from Ben Franklin, who said, "Who hath deceived thee so often as thyself?"
That's perfect.
Awesome. I love it.
I mean, it's so because viruses, bacteria, you can understand they accidentally get in and they're causing issues, and they have evolved to escape a lot of these things. But ourselves, It's so weird, right?
Right.
Evolving to escape ourselves within ourselves. It's very philosophical too, if you think about it.
Yeah.
Yeah. And so it's really fascinating. So this idea that cancer cells are self, how would you recognize a cancer cell as a cancer cell? Do you have any ideas?
Are you asking us?
Yes. How do you recognize a cancer cell as a cancer cell if it's self?
Well, if it has enough mutations that it's able to then express certain antigens that are different enough, I think would be the answer.
That's right. So those are called neoantigens. So those are antigens that are unique to cancer cells. Another way they can do it is they can upregulate proteins that are normally only expressed at a particular time during development or in a very specialized location in the body that's immunoprivileged. And so there's things like testes antigens or developmentally regulated antigens that the tumor cell upregulates just because they are so proliferative and enzymatically active, and they somehow upregulate these molecules, so they're at significantly higher expression than on host cells. Another place is in melanoma. So melanoma has melanin and a bunch of other proteins that are expressed uniquely in those types of cells, and so if you can generate immune responses to those, you might lose, in my case, freckles, or you might lose pigmentation in your skin, but you could eliminate the melanoma. So people have been using those antigens to try and drive immune responses. And so the idea here is overcoming tolerance. So the tolerance to self is in place normally. And so if you can overcome that tolerance, you could generate an immune response.
Let me ask you a question about PD-1 ligand.
Yes.
So PD-1's on the T cell, and then the tumor cells produce the ligand, and
that's right.
That's how they put the T cells to sleep.
That's right.
I assume other cells also produce PDL ligand, PDL1, right?
Yes. So I don't have a whole list of those, but certainly other cells can upregulate that. One of the things that upregulates these are inflammatory stimuli. So anytime you have inflammation, you have upregulation of things that are going to dampen that down. And defects in that process contribute to chronic inflammatory diseases as well.
Would also a cell like a neuron that doesn't want to be lysed by a T cell, would they have PDL1?
I don't know, but I would think so.
It would make sense. So, so in a sense, the cancer is not doing anything new. It's just, as you said before, it's expressing when things should not be expressed. But there is the potential to do that elsewhere. Okay,
that's right.
Yeah,
that's right. So, so there are also some autoimmune diseases that are associated with PD-1 and polymorphisms, like lupus and type 1 diabetes, rheumatoid arthritis, Graves' disease, multiple sclerosis. And so there's this idea, perhaps, that some defect in this pathway of T cell regulation through these molecules could contribute to, regulating the immune system, and maybe this would have potential for tumor immunotherapy or cancer immunotherapy. And so one of the hints for the potential of this whole system to work is that people found high levels of PD-1 on PDL1 on tumors, and the higher the level of PDL1, the poorer the prognosis for a bunch of different types of tumors. So we have these hints. We know how T cells are regulated. We know that the tumor cells are upregulating this ligand. We know that the ligand is shutting down the T cells. So there's all these hints of how this might be working in the system. And so the last piece of information that we need is how the T cells are actually killing a cancer cell. And so if you have these cytotoxic T cells, they can recognize these neoantigens in the context of these MHC molecules, and they can deploy their normal cell killing mechanisms, and these things are called perforin and granzymes. So perforins perforate the membrane. Granzymes are enzymes that go in and they cause the target cell to just undergo this death process, this programmed cell death. So other cells that can contribute to the cancer cell death are CD4 T cells that are helper cells, and they secrete cytokines. And those, when they recognize their antigen and they secrete cytokines, they secrete things like tumor necrosis factor and interferon gamma. And those, as you can imagine, have an effect on cancer cells that are going to help to kill those cells. So we sort of already talked about why it's challenging to attack these cancer cells and what these T cells are capable of, but how they're suppressed. So we sort of have the players in place now. So exactly how did we get there? What is the history of the development of these immunotherapies, given those bits of information? And I think one of the most fascinating aspects about this is a very recent discovery in evolutionary times of immunology, right? So the early 1980s is when we started to understand how T cells had specificity and what T cell receptors were and how T cell receptors engaged with MHC molecules and how those signal 1 and signal 2 activated T cells. So really, that's not a long time ago. It can take that long to develop a therapy and get it to market. Right? This was when the initial observations happened. And in the late 1980s is when a molecule called CD28 was identified on T cells. And that's that molecule that receives signal 2 to let the T cell know that its T cell receptor is seeing something important. So what are these molecules? I'll just remind everybody that in Immune 4, when we talked about CAR T cells, we talked about CD28 as being one of these molecules that activates T cells. And so if you're interested, there's a little bit more about that back in that one. But the CD28 molecule, and by the way, CD cluster of differentiation, it's just this nomenclature in the immune system that's just crazy. But it's a molecule that's on the surface of T cells that helps them receive this, co-stimulatory signal. And so we knew by then how to activate T cells, but we didn't know how they were shut off. And it was assumed for a very long time that they just died once there was no more pathogen around, there was no more antigens being presented in MHC molecules, and the T cells just sort of withered away and died. And it turns out that that's not the case, as one could predict by what we were already been talking about. And so in the early 1990s is when Jim Allison discovered the CTLA4 molecule, and that this molecule engaged CD28, but shut CD28 down. So normally CD28 provides this positive signal 2 co-stimulation, but CTLA4 engages that CD28 and prevents it from signaling.
Do you remember how he discovered it? Was it with an antibody?
Oh, I'm not sure.
I mean, I would almost guess that, right?
Yeah.
That'd be the easiest way to do it.
I know that once they understood how it was activating the T cell, they were able to make humanized antibodies to knock it out. I think they originally made antibodies to it and showed that if they blocked it, they could block T cell shut off.
Right. Makes sense.
Yeah. Yeah. And one of the other ways in which they showed how this is involved is what immunologists did then and still do is make a knockout mouse, right? So they make a knockout mouse of the CTLA4 molecule, and those mice died very early in life. They survived through birth, some of them, but they had aggressive autoimmune inflammatory disorders. And so that, really cemented the idea that without this molecule, the immune system was unleashed. And so in 1996 is when really there was the hammer in the coffin, if you want to say that, for cancer, is that they made this antibody to CTLA4 that would block the interaction of CTLA4 and the T cell, and this antibody in a mouse cancer model cured cancer. And I want to go back because I made a mistake. The CTLA4 doesn't bind to CD28. CD28 and CTLA4 compete for the same molecule. Sorry. I messed that up. But so he made this, and they published this paper in Science in about 1996 where they showed that this antibody was immunotherapeutic in cancer. So they could take the brakes off the T cells, and the T cells would go and kill the cancer cell. And so that was just on this timeline again, this was 1980 to 1996 was the mouse model. By 2000, the first clinical trials showed that there was response in patients.
That is fast. I would imagine heavily motivated by the potential profits because of cancer being one, like you said, 1 in 4. But it is short. I don't think the public can, it's hard to imagine how fast should things go, but in science, that's really fast.
I remember reading that he had trouble getting a company interested because they said, it works in mice, who cares?
Yep.
Right?
Yep.
So he had some trouble to get it done, but eventually he persisted and got it into people.
Yeah, so his original studies were done at Berkeley, and then he moved to New York City, as you mentioned, to work at Sloan Kettering so that he could try and develop this therapy. So that was one of the motivations for his move there. And so they were really interested in aggressively trying to develop this. And so one of the cool things was that first trial in the 2000s was a phase 1 clinical trial. And we can talk a little bit more about what phase 1 and phase 2 and phase 3 are in a little bit. But this phase 1 trial, which is just literally you take people who are really, really sick, and you're not sure if you can make them better, but you certainly can't make them much worse. And you just inject things into them and you see if they tolerate it or if it, has toxic side effects. And even in this first clinical trial for this phase 1 with just a few patients, they saw full clearance of tumors.
Wow.
I'm
imagining that does not happen a lot.
That does not happen a lot. One injection of this immunotherapy against CTLA-4, and they had a patient that still is about 15 years now, 18 years out, with no recurrence.
Wow.
It's amazing, like absolutely stunning.
So what was the type of cancer that they were treating again?
Yeah, metastatic melanoma.
Okay, metastatic melanoma. Got it. That's important when we talk more about the types that later on, how checkpoint inhibitors can work for different people and different cancers. I think that'll be important down the line. But it's fascinating. And, we can go back. I don't think you said anything wrong about CD28. I think we can talk about it being a competitive inhibitor, but I think you were fine.
Okay.
I think it's, I was trying to go back.
I just realized I thought it was. It doesn't matter. I thought I made a mistake in what I said. No. But it's okay. Dare I be wrong, right? I'm wrong all the time.
Yeah, so we all are, but we're glad we're glad you care enough to fix it. That's
exactly. So it makes a good scientist, right?
Right.
Yeah.
So if we continue on in this timeline, the first checkpoint inhibitor antibody that was approved by the FDA in 2011. So again, now, 1980, we discovered T cell, receptors. The late 80s was the on molecules. Early 90s was the off molecules. 2000 was the clinical trial. And by 2011, it's approved. And so this is called ipilimumab, which is known as Yervoy. They always have these weird names. And so I would suggest people go back again to Immune 4 again, where I talked about what's in a name, my pick of the week that week, because it's interesting to know what the different parts of the molecule, what the name mean. And obviously MAB is monoclonal antibody. It's really easy. But I thought so again, I was digging stuff up for this, and I thought one of the funniest things I heard Jim Allison say in a talk is like they were originally calling this thing M something 101. And so when they went to do FDA approval, they had to give it a name, and they said ipilimumab, and he's like, that's a terrible name. And he said they actually should have put an H in front of it to honor the origins in Berkeley, and it would have been ipilimumab or whatever. So like hippies. But I thought that was really funny. But so this ipilimumab is the first one that was approved, and that was 2011. And in 2014, there were like 2 or 3 more that were approved, and now there are a whole bunch of these things.
For both molecules, or are we just on?
For both molecules. Yeah. So targeting. So there's actually 3 molecules. There's targeting CTLA4, which is this ipilimumab, and then there's targeting PD1, and then there's targeting PDL1. So you can target any one of these 3. And I think what's really important to mention at this point is that this idea of targeting a molecule on the immune system cells is a massive shift in how we think about treating cancer. Because if you think about what is the standard of care or was, or mostly still is surgery, chemotherapy, and radiation. And every single one of those mechanisms is targeting the tumor. Let's cut the tumor out. Let's kill the tumor cells. But we're not thinking about the immune system, right? So there's a little bit of change in that with some of the cancer vaccines that people were trying. They were taking the cancer out and then chewing it up and then trying to inject it back in and get the immune system to attack it. So this idea of cancer vaccines. But now this is a complete shift in the way we're thinking about this. And we're saying, like what we started by saying is like, there are these cells in the body, these immune cells that are capable of attacking the tumor, but they've been shut down. And can we reinvigorate them? Like, like a zombie, like bring them back to life and have them attack the tumor. And so that's really the shift in what's happened with how we think about this immunotherapy. And so, so this ipilimumab was to CTLA4, and then there's PD1 antibodies, and that one's called pembrolizumab or Keytruda. There's another one called nivolumab, I can't say these, which is Opdivo. Now that one I've seen a lot on, commercials on TV or whatever. And then a third one that I feel bad because I'm not sure I can pronounce it, Semipimilab, Libtayo, I don't know. But one of the cool things, and then there's PD1 antibodies, and so people might recognize these if their family members have been treated with these. So PD1 antibodies are atezolizumab.
PDL1, like the ligand instead of...
Yeah, so these target the ligand. And then there's avelumab, which is also known as Bavencio, maybe that's Italian, I don't know. And then there's durvalumab, which is Imfinzi. I don't know. These sound Italian to me. They all have lots of K's and Y's and Z's and Immies and whatever. I don't know. I think it's really fascinating. But so this Opdivo, which is this nivolumab, which is targeting PD1, I thought this was really cool because we've talked about humanized antibodies and bifunctional antibodies in a previous episode and things. So these humanized antibodies, the problem with antibody therapy that you need to think about is like if we make an antibody in a mouse or a goat or a pig or whatever you choose and we inject it into a human, it's non-human enough that the human will make an immune response against it like it's a foreign molecule. So you're really limited. So you could inject someone with antibodies that were made in a goat against human PD1 one time because the immune system will generate a response. And if you try and inject them again, the immune complexes will just be cleared and there will be no functional antibody around anymore. So you need to make the antibody as if your own body is making it. So you have to make it fully human. And I thought it was really cool because they did this in a different way that I didn't realize people did. And that is they humanize a mouse. So they make the mouse express the human immunoglobulin locus. And then they immunize the mouse with the human PD1 molecule. And then they were able to make a fusion for a monoclonal antibody, which we can talk about how that's made at another time. But they make a monoclonal antibody from this mouse that's actually human because they made the mouse human.
Right. So that gets around humanizing the
That's
antibody. Right. And the problem there is that it's got patent protection. So, it's not so hard to humanize an antibody, but a company that wants to make a product say, we don't want to deal with this patent. So they made a mouse. That's probably the driver, I would guess.
Probably. Right. But I just thought that was a really cool way of doing it.
Yeah.
Yeah. And so another interesting part of this is all of these are going to have different isotypes and different affinities and different, whatevers of these antibodies. And this particular one that I'm talking about, this Opdivo one, is an IgG4 if you follow these kind of minutia of the isotypes. But it's interesting because they picked that one because it does not activate complement. And we talked about complement in Immune 7, about Alzheimer's disease and things. So if you're interested in complement, you could go there. But it's interesting that you choose the isotype because you want certain functions and not other functions. And so activating complement would probably not be advised in this kind of scenario. So they didn't want complement activation, which I thought was cool. So when they tried this clinical trial with this nivolumab, it showed a 40% objective response and a 72.9% overall survival rate. I mean, compared to chemotherapy, which had a 13% response and a 42% overall survival. And I don't remember, I forgot to look at which cancer type that was, but a lot of these have been tried in melanoma. But that's pretty amazing. Like you're almost doubling the overall survival rate. These just seem like miracle drugs, right?
Yeah, for sure. So that brings up a question: is like, what's it worth? How much would you pay for it? Right? Because we talked about CAR T cell therapy, which is
$300,000 or something.
Yeah, and the company said, "Well, it saves your life." What is it worth to save your life? Well, that's not the problem because no one can afford. I mean,
right?
Well, sure.
Us, we can't just shell out $500,000 for a drug, but insurance companies could. It's up to them to approve it, right?
Yeah. Well, and it's flawed logic to say what's it worth your life, because then you're putting worth on socioeconomic barriers of people. It's not. Do that. It's icky. Yeah. Very icky.
No, so while you guys were talking, I looked this up, and unfortunately, it's not much better. So, so each inhibitor on its own is about $150,000 a year, and the combination of the PD-1, I think, and PD-L1 that works really well is $256,000 a year. So you need to get in a clinical trial.
Right. And, I think we do talk about this in previous episodes, but, in defense of the drug company, there's the amount of money they're spending to run these clinical trials, to test and bring it to market. They would say that warrants that. And I would imagine the only way to make this better is just more competition, but, more science to make there's more availability, I would imagine.
I also think the government should play a greater role in funding. Right. I mean, I heard a wonderful podcast with a economist in the UK whose specialty is pharmaceutical economics, right? And she said that the companies always say we're repaying our research. She said, but that's baloney. They just want to make as much money as they possibly can.
Right.
And her thing was that the government should fund a lot of it and then get some back for its investment. And that way the drugs don't have to be paid. Nothing has to be paid by the people who get it. Because in the end, really, it's the insurance companies who are going to decide, we'll pay a certain percentage of this. And then if it's a lot, they'll raise your premiums, right?
Right.
But that's not the way it should work.
Right.
That sounds interesting. Could you post that? If you find that, could you post that in the show notes?
Yeah, I think it was a pick on one of my podcasts. I have to find it. But yes, it was. She was amazing. She was so good. And she just ripped off these arguments, why the government, she said almost every drug made in this country, FDA approved, has some NIH funding, at least to start. And she says the government gets nothing back for that. She said it's crazy. If it were a company, you would never give away. So they take nothing, and they should, so they can invest in more companies, right? They should really have that. Right. And then there was her whole argument about recovering research costs, which she said is just nonsense. But I will find it and put it in the show notes. Yeah, it's got to be somewhere.
Cool.
But that's so true, though, because you would imagine that Jim Allison was primarily funded by NIH for all those initiatives.
Yeah, for sure. So, and the government's getting nothing back.
Nothing.
Yeah. It's
except a bill to cover costs of therapy.
Yeah.
Yeah. So continuing on, one of interesting things here is so we've talked about CTLA4, PD1, PD-L1, and you can target any one of those. But my brain started to explode when I started to look at these newer images with all of the checkpoints on them. There's like 20 different ones. And so it's interesting because each one is going to have slightly different impact. And so how many of these do we combine together? So one of the problems with, for example, ipilimumab treatment is it works great when it works, but it only works in a few people.
Right.
And
Right.
so trying to understand why, what is special about those individuals? Why do they respond and the other people don't? Why is it only 20 people respond or 20% of the people respond? And so that's a key to understanding where we go forward, right? And that's part of the idea of combining these things together, because if only 20% of the people responded to PD-1 therapy and 20% of the people responded to CTLA-4 therapy, what happens if you put them together? And in fact, I was watching a talk that Jim Allison gave, a scientific talk, where they titrated things down in the mouse where they could use anti-CTLA-4 and have no effect on the tumor. They could use anti-PD-1 and have no effect on the tumor. But if you use those two together, every single mouse eliminated the tumor. And so there's some sort of synergistic effect between those two. And what the proposal is that CTLA-4 blockade is really good at letting the T cells get into the tumor, but most of the people's T cells that get into the tumor get shut down right away from the PD-L1. So they have high PD-1 on them. So even if they get in, they can't do the job. On the flip side, the ones that the PD-1 blockade or PD-L1 blockade work on, the blockade works great, but there are very few T cells that get into the tumor. And so if they use those two together, what they found was that the T cells got into the tumor and they got activated. And so it's this idea of really understanding the basic biology of how each one of these checkpoint molecules functions, so then being able to rationally decide which ones at which time and which patients you want to target, I think is really interesting.
Yeah, I think the dream would be is if for every cancer type, we would know the neoantigens that they produce, so the specific antigens that are not recognized as self, that then, okay, what can we treat with that? What combination of checkpoint inhibitors? And based on some of the clinical results, you're right, if only 20% of the people are responding, okay, we may need there might be a day where we're looking at people getting 8 to 10, it could go up to maybe 20 different checkpoint inhibitors to really maximize. Because they say that, melanoma is really responsive, but like colon cancer is not.
It's really not. Yep.
Right. So I mean, that would be so neat. But going back to what we talked about, the drug companies do not necessarily have a financial incentive to share that information. So what would make them talk to each other?
I think it's advocacy, right? It's patients advocating and things like cancer advocacy programs to make these things happen.
Yeah, I mean, organizations that, talk for patients, American Cancer Society, hey, you need to do this, right?
Yep.
Put some pressure. Government, FDA, I don't know how it works because I'm not in pharma, but yeah, you would think that something has to be done because there have been combination trials, right?
Mm-hmm. Yes.
And so
it's working.
Someone got that to work. And,
right.
there's a lot of activity in using viruses to kill tumors, as you know, and now that's being combined with some of these checkpoint inhibitors.
Right.
Because it works even better with them.
That's right.
So,
the reason why is because when they lyse those, they're called oncolytic viruses. When they go in and they infect the cancer cell and they cause the cancer cell to explode, basically, they're releasing all of the antigens and all the danger molecules, causes inflammatory reaction, and that helps to activate the immune system and release those antigens that can then be presented.
And that is what one of our listeners wrote in, is the abscopal effect.
Yes.
He said you can lyse a tumor in one part of the body and it will help get another one somewhere else because of all this activation of the immune system, the virus, the release of antigens, and if you include these checkpoint inhibitors as well, they're all moving around, so it's not restricted. Even if you injected virus into a tumor, if there were another one somewhere else, it could be affected. So,
yeah,
very cool.
Yeah. So I just wanted to emphasize one more time this timing here. So, we went from initial discoveries to marketing a drug targeting, this basic discovery in 15 years.
Mm-hmm.
And then the Nobel Prize was awarded 7 years later.
That's pretty fast.
It's really fast.
I mean, some discoveries that happens, right? It's really quick. Right. Reverse transcriptase is another one that happened really quick after its discovery. And sometimes I think it's really clear that something is really important, right?
Right.
And other times it takes years for it to come together and you look back and go, oh, yeah, that was important. Let's give him a Nobel Prize for that. Yeah. And it may take a long time. Peyton Rouse discovered Rouse sarcoma virus in 1911, got his Nobel Prize in 1960.
Is that right?
It took him that long to realize what he had done. Yeah. Nobody could put it together. The whole cancer history is so muddled that it took forever to put it together. And then he died shortly afterwards, so at least they got him, right?
Yeah, exactly.
Yeah, I think that's cool when it happens really quickly. I wonder how long CRISPR will be.
I don't know. I was wondering if that was going to be this year or not.
I was thinking so too. I think it'll be fast because it seems to be people are making a very big deal about it. It doesn't seem to be they have to, wave the drum roll and try to get attention.
That will be a controversy because
it will.
There was a whole history before Doudna and Charpentier and Fang. And then there are other people, so they can only give 3, as you said, and so who's going to get it? I mean, I can't see them not giving it to Doudna and Charpentier, but if you don't get some of the early people, that's not quite fair, right?
Yeah.
On the other hand, the utility for gene editing is really Charpentier, Doudna, Fang, so I would predict those are going to be the 3.
Probably.
Yeah. And I have no insight.
I don't. I'm not on the committee, so I don't know.
No one's talking to you.
No, I don't have an inside lead. No. So there were like a couple of other things I wanted to touch on, and one is that people who might listen to this might have a family member who would have this kind of therapy. The question is, how does that actually work? How do you deliver something like this? If you deliver rabies immunoglobulin to someone who has an exposure to rabies, is an intramuscular injection of immunoglobulin. But in this case, it's an IV injection. It's an infusion, so it's basically just like getting chemotherapy. And, all of the timing, frequency, dose, how often you get it, how long you get treated is still sort of an art. There have been approved protocols, FDA approved uses, but, there's so many clinical trials going on testing a variety of different combinations and timing and whatever. And there's so many different cancers. It's approved for sure melanoma, lung, Hodgkin's lymphoma, and it's being tested for probably hundreds more types of cancer. So, it's still very much in flux as to how this all works. And when I looked it up, there are currently over 1,500 clinical trials currently going on, testing various different combinations of checkpoint inhibitors. And just contrast that. So that's as of September 2018. Contrast that to 2016, there were 215. That's how much this whole field is exploding that I think is really fascinating. So, yes, it's $300,000 for treatment, but maybe you can find yourself a clinical trial to get into. Right. And some are trying this without previous chemotherapy. Some are trying it before you start surgery and chemotherapy. Some are adding it on as combination therapy or adjuvant therapy, as it's called. Some are adding it on after if you failed previous chemotherapy. So there's all different potential options. I think that it's really interesting to note that for especially metastatic melanoma, it seems to be that's really the one that this works well for, because metastatic melanoma has a really poor prognosis. There's a median survival of like 11 months, but some are much, much shorter than that. And then there was a phase 3 trial with these combination therapies and things that saw 22% the people still alive after 5 years. It's really just it's amazing that you can have that long-term survival rate.
Must get rid of all the cancer cells, right? Because usually you don't, and they come back. Surgery, you don't get them all or whatever. And so this is clearly wiping them out because it's you. It's your immune system, right?
So it could be, or the other possibility is that even if there's some residual cells that continue to proliferate and move around the body, those T cells are already still primed and they've overcome that tolerance so that if they see their antigen again, they'll kill it. So just like if you have an infection or you have a vaccine, those memory cells stay around for a very long time.
But why aren't they susceptible to PD-1L then?
So that's a great question. Maybe the blockade causes downregulation of the molecule on the surface. I don't know if they upregulate it again later. I'm not sure.
Yeah. You may have mentioned this, but I don't remember. This is also being combined with CAR T cell therapy, right? You put the antibodies with CAR T. And I seem to have read it somewhere that people are looking into the combination. Does that make sense?
It would make sense.
It would make sense.
Because not the CAR T, you're directing them towards an antigen, and then here you're taking away the checkpoint. So both might even do better, right?
Probably, because remember CAR T, what you're doing is you're targeting the T cell to a cancer antigen and then giving it the co-stimulation. But still, if you have the co-stimulation, but then you have a shutdown signal, you could still interfere with the ability of that to kill the cancer cell. So yes, I would think that would be irrational.
And thinking about the ability of it to work, if you can't get the T cell to infiltrate, and I think that's one of the correlates of the ability to kill the tumor cell, if you can't get it to the tumor, you're not going to be able to have a good prognosis. So I think that, and I'm looking it up, and there's a lot of combinations. And if anyone out there is working on that, you can write us and talk about your exciting work, and we'll read about it.
So can these antibodies get in the brain, Cindy?
Antibodies don't get in the brain very efficiently. The blood-brain barrier is pretty good at keeping them out. I know some can under some circumstances. I'm not sure if it would be a good therapy for glioblastoma, for example, if that's what you're thinking.
Yeah. Could you inject? So they're injecting poliovirus into glio, but could you inject antibodies in?
I would think so, yeah.
Yeah.
I guess you have to make sure that there is a good infiltration of T cells there too, or call them in somehow. But definitely the T cells come in when there's issues going on in the brain for sure. So yeah.
Although maybe that wouldn't be good, right? Depends.
Causing that inflammation in the brain could be problematic. Yeah.
Right.
Yeah. Okay.
So that is one of the problems with this therapy, right? So it causes a lot of itises, a lot of inflammation all around the body, and so it can often, believe it or not, cause the cancer to grow first before it goes away, because cancer cells like inflammation. They like some of the factors that are produced. It helps them grow, and so if you pull the brakes off the T cells, you can have this systemic inflammation, which has a lot, it's contributing to a lot of these side effects that come along with this kind of therapy. And the worse you get the side effects, the higher the chance that you eliminate your tumor. So there's also a balance there, right? So you have to fight the side effects so that you can survive the therapy. But if you survive the therapy, you get rid of your tumor, probably. So yeah, it's not ideal, but when it works great. Right? Yeah. So the other thing I came across that I thought was really neat that I'd like to mention is I thought that tumors also get really smart this way in that they shed little bits of themselves, these little exosomes that they put the PD-L1 the PD-L1 on. And so those little shed exosomes can shut down the T cells without the T cells actually have to contact the tumor cells. So they can shut down those T cells wherever they are in the body. So the primary tumor, for example, could help protect its babies as they metastasize the body from the T cells, because as the T cells are circulating in the blood, they might get shut down by these exosomes. I thought that was really cool.
And I would think if you are the proteins on the surface of these exosomes, or the inside? They're on the surface. So if you're using an antibody to PD-L1, that could be a problem. The antibodies would go after those, right?
Right. And maybe that's why it doesn't always work.
That's right. So that was the paper I read was talking about that, where they said that, the one of the things that correlated with failure of therapy was these high levels of these PD-1 positive exosomes in patients. And so they were arguing that that's potentially a biomarker. So if you have metastatic melanoma and you survey the blood, you could look to see if they have lots of these exosomes, then the therapy might not work, or you potentially have to just inject a lot more of the antibody to overcome the kind of the sink effect of the exosomes sucking up all the antibodies that they don't work right.
Well, and I think, that would be a good biomarker to be able to know how much do we give, how much do we fine tune it? Because you're right, then more could be a worse autoimmune response or inflammatory response as well.
Exactly. So you might have to tweak it in each patient, but there's just a lot. There's a lot going on. But so these combinations that they've been able to put together. So, maybe if your exosomes have PD-1 or PD-L1, you could use combinations where maybe not all the exosomes have those molecules. And so there is one example that I found of a phase 1 trial where they used antibodies to CTLA4, PD-1, and PD-L1. And again, phase 1 trial for metastatic melanoma. Remember, it's typically 11 month survival. 88% of the patients in that phase 1 trial were still alive 2 years later. And it's like this is crazy that this stuff works that well. I mean, the chemotherapy does not work this way.
No. And it's wild that it took, I don't know if it took evidence to have this change in attitude towards immunotherapy. Because like you said before, treating, I don't know, they used to treat with trying to do interferon-based treatment with tumors. And chemotherapy, it's such a harsh, non-specific way. So it's nice that really, I know, Vincent, we're biased, but the immune system, it's where it's at. If we can learn and understand all of the nuances of the immune response, we could treat anything. That's the dream, I think.
That's the dream for sure.
Speaking of treating anything, what else can you do with these, Cindy?
That's a great question. Yeah. So I thought
Oh, someone's popular.
So I thought this is really cool because these are getting the spotlight for treating cancer, right? But these kind of idea of regulating T cells important in many different diseases. So in autoimmunity, rheumatoid arthritis, and systemic lupus erythematosus, those T cells are too active. So can we shut them down? So could we engage with agonistic antibodies? So antibodies that activate the molecule instead of block the molecule to shut down the T cells. In infectious diseases like chronic viral infections, so we can talk about viruses, maybe those that tend to silence those CD8 T cells because they get this phenotype called exhaustion. So those T cells are working so hard to try and eliminate the viral infection, but it's a chronic viral infection. You're never going to fully eliminate it. So these T cells just become exhausted. They just run out of steam. They're in a marathon and they reach mile 25 and they just can't finish the race, right? So is there a way to reinvigorate those T cells by targeting, again, these PD-1, PD-L1 molecules to release the exhaustion phenotype? There's also parasite infections that tend to suppress or block T cell responses through upregulation of these molecules that shut down T cells. So could we use them perhaps in things like, I'm not sure how we say this, Tinea cresceps? Somebody will.
Tinea cresceps.
I'm glad you know it from being on
TWIP.
There you go, TWIP.
And I know how to say schistosoma mannsoni. So those two I found have blockade of T cell responses. And so if we could target potentially these PD-1 and PD-L1, we could release the inhibition and allow the immune response to try and eliminate some of those pathogens. There's also in transplantation. So in transplantation, there's upregulation of these PD-1 and PD-L1 molecules in transplant recipients. And if you block it, it enhances rejection. So this is a situation where maybe we could agonize, or agonize seems like a weird word, but activate the PD-1 molecule on T cells to help keep the transplants and prevent their their rejection. And the last thing I found was in allergy and asthma. And so this is interesting because the PD-L1 is the molecule we've been talking about, but there are in fact two members of that family, PD-L1 and PD-L2, and they can both engage PD-1. And so in asthma, there seems to be PD-L2 may play a role in blocking, if you block that, it's blocking increased airway hyperresponsiveness and eosinophilia. So perhaps if we are releasing that, we might help to reduce those hyperinflammatory responses.
Are any of these in people yet?
I don't think any of these are actually in people. All I saw was preclinical, but that doesn't mean there's not a clinical trial currently going. I would think if any are targets for initial interaction, it probably would be autoimmune disease or chronic viral infection would probably be the first two places people would try those, but I'm not sure.
Neat.
And I don't know if we might not have time to get into more mechanistic conversations, but I did have a question regarding, because CTLA4 is expressed on not just CD8s and CD4 T cells, but also on things like T regulatory cells, which, if I'm thinking about it, would work against, if you're blocking CTLA4, I was looking at it actually increased it increased T regulatory cells. So I think there's probably not an answer for this, but it's not going to be T cell specific all the time to CD8 T cells, correct? I mean, it's going to hit other subtypes. And
it will. Yeah.
How to understand that? It's just kind of a weird way to think about it because increasing T regulatory cells would go against activating CD8s.
It definitely would. And I think it's, again, coming back to that rheostat idea, you might be enhancing T regulatory cells, but yet if you get enough of those active cells into the tumor and you can activate them, even with some regulatory T cells there, it might be enough to overcome the tumor.
Sure.
But certainly thinking about, should it be combined with something like try to eliminate T regulatory cells and then use this therapy? But there's probably issues with trying to eliminate T regulatory cells that could cause
and maybe
other problems.
Right. Maybe that's why the combination is required in the patients it works great, but then you might need it. And maybe that's the immune system's way of saying, okay, just one thing isn't going to change the way I behave. I need 2 signals, 3 signals because of the layers of checkpoints. So it's interesting.
Yeah.
Well, Cindy, that was great.
That was awesome.
Love it. Thank you so much.
It was really fun.
It was really great through the whole story, explaining it. I'm sure our listeners will love it.
Yeah.
We appreciate it.
Pairs well with the CAR T cell episode. So if people listen to that one first, then this one, you get a lot.
So, by the way, Cindy, when you teach and then you get a Nobel in immunology, you must bring it in a classroom and get excited about it, right? Do they like that?
I do. I talk about it. Yeah. So
have you taught this yet?
Funny. I did. That's the funny thing about the timing of all of this. I just taught, and so shout out to my veterinary students. I just taught them on Monday immunoregulation.
Oh, really? This past Monday?
Yeah.
Wow.
This past Monday. And I talk about a bunch of different things, T regulatory cells, soluble, pro-resolving mediator, lipids, and a whole bunch of other things. But, we talk about IL-10, TGF-beta, cytokines, but I also talk about surface molecules like PD-1, PD-L1, and CTLA-4. And I mention the Nobel Prize, and I'm like, this is why you guys should care. And people perked up a little bit. It's hard to teach them 9 o'clock in the morning. They don't want to be there. But when I mentioned, there was a Nobel Prize, and I was like, did you read about that? And they're like, of course not. We were in class. It's a veterinary school. Always cool. Study, but it's fun to bring that into the classroom. Especially undergraduates love that too when you talk about that. And my pick of the week, although we're not quite going there yet, is a list of all of the Nobel Prizes that have been awarded in the area of immunology, because I think it's a timeline of how we discovered the immune system. And it's just amazing that each major finding that we have in helping us understand how the immune system works has, almost all of them have garnered a Nobel Prize at some point in time. So as we make new major shifts and discoveries in immune system and how to exploit it, it's more and more and more Nobel Prizes. And that particular website hasn't quite updated yet, so they don't have the 2018 on there, but they have the TLRs on there from 2011 and 12. So I think it was 2012. Yeah.
Well, we could jump to picks of the week. We could do emails another time, and
yeah, it's okay with me.
Okay, that's
so that site is immunologylink.com/nobel.html, and so it really just lists the Nobel Prizes that were given to discoveries related to immunology.
Some big names here.
Yes, many big names.
First one was 1901.
Yep.
Now, I recognize Karl Landsteiner, 1930 blood groups, but he discovered polio in 1906.
But he didn't get it for that.
Didn't get it for that, no.
Groups.
No, it wasn't the first human virus, so.
Yep.
Susumu Tonagawa. So I was at MIT and he moved to MIT, and I used to see his office out my lab window. I used to watch him sitting there at his desk. And who knew he would get a Nobel Prize? Yeah, monoclonals. So the last one before the current one was 2011, Beutler and Hoffman, yeah.
Beutler, Hoffman.
Steinman.
And Steinman.
Cool, this is a great list.
Yeah, it's fun.
Very nice. Steph, what do you have for us?
Yeah, there is a website called immunobites.com, and it was highlighted. I found it on Twitter. Akiko Iwasaki from Yale, she had retweeted it, and I love it because it's a SciComm project, and basically it's a group of what looks to be PhD students. I'll have to go back and look. I don't know if there's postdocs, but PhD students who are writing these little blurbs of immunology on a paper or a concept. I think if you go to the website, the most recent one is somatic recombination, and they are also have a call out if you're interested in writing and learning how to write. They would be happy to take your name, I guess, or application. You can go to their website, but it pairs well with what we're doing, trying to communicate immunology. We're doing it, with audio, and they're doing it visually with writing. So I just wanted to raise them up a bit.
So it looks like there are a couple of PhD students, a postdoc, and a technician as well.
Yeah, it's great.
Cool.
I love it.
Yeah, these are good. There's another one at Michigan. Same idea. People who want to write, they get together. And when you have a few people, it's easier. So you don't have to come up with a post every week. Very good.
That's true.
My link is to Brianne Barker's immunology course videos on YouTube. So
Yay!
Brianne is a recent joiner of TWIV, and she's a professor at Drew University over in New Jersey.
She's great.
Teaches immunology and virology, and she got inspired by me posting my virology course videos on YouTube. So she's doing the same thing for her immunology course. So I think that's great, and there's nothing better that I feel I can do than to inspire other people. So I think that's cool, and it's also a good immunology course if you want to take one. A lot of people ask me on YouTube, is there an immunology course? So there you go. Brianne Barker.
Oh, good job, Brianne.
Yeah, great.
Congratulations. It's awesome.
And that will do it for Immune number 14. And we will have one more this year in 2018, right? December.
Yes.
You can, I'm sure everyone listens on a phone or a tablet. You have a favorite app that you listen on. So first, we'd like you to subscribe using that app, and that pushes our numbers up. It helps us to try and get support for the show. And if your app allows you to favorite episodes, please do that. I use an app called Overcast, and each podcast episode you can actually hit a little star and favorite it, and that makes it rise to the top, so that if you go in the app for recommended podcasts, then you can see, some of ours. So we'd like that too. And, of course, questions and comments. We'll do some of those next time. Immune@microbe.tv. And finally, if you really like what we do, consider contributing financially to all of our shows at Microbe TV, not just Immune, but all the others. Microbe.tv/contribute. Cindy Leifer's at Cornell University. You can find her on Twitter as Cindy Leifer. That was great, Cindy. Thanks so much for putting the work into that.
Thank you.
Yes,
it was great.
awesome.
It was so fun.
Really
I
good
love
stuff.
doing a deep dive in something like that. It's really fun.
Yeah. Steph Langel, for now, is at Ohio State University. She's on Twitter as Stephanie Langel. Thanks, Steph.
Yeah, thank you. Thank you both. It was great.
I'm Vincent Racaniello. You can find me at virology.ws. I am Prof VRR on Twitter, YouTube, and Instagram. PROFVRR. By the way, go to YouTube, youtube.com/PROFVRR, and subscribe. I need 25,000 subscribers to be able to apply for YouTube educational video grants. So it's free and you might like the content. youtube.com/PROFVRR. We are at just shy of 17,000. Come on, folks.
We need to get you a viral video. I think that could really help drive subscribers.
Totally. I totally agree.
Totally.
And believe me, I have tried. I have so far failed.
I'll try to think of something.
But I do not give up. One thing about me is that I am persistent. And so I am going to get it. So please do
I
that.
subscribed, so there you go.
Thank you. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.